Okay, everyone, so how to clone a DNA fragment in molecular genetics. In other words, how to make a clone. So let's assume we are starting with a DNA fragment, and the DNA fragment could contain a gene, it could contain part of a gene, it could contain an intergenic sequence. You know, it doesn't matter. And let's not worry about how we got the DNA fragment. I mean, we could have gotten it by PCR, we could have chopped up genomic DNA and we have a random collection of DNA fragments. But let's not worry too much about that. Let's just assume we have a DNA fragment and there are Hindi3 sites, Hindi3 recognition sequences close to the ends of this DNA fragment. These are the worst pencils. You know, if you sharpen them, they go dull fast, and when you sharpen them, they always break. Okay, so first thing we would do is digest the DNA with Hindi3, with the Hindi3 enzyme. So the DNA is in a solution of water, we add a buffer that is compatible with the enzyme, and we add the enzyme. And the enzyme will cut the Hindi3 sites on the ends, and digest is just a word that we can use interchangeably with cut. So cut the DNA or digest the DNA with Hindi3. And what will that do? Well, that will give us a DNA molecule that I'm going to, diagram like this. So a DNA molecule with sticky ends. So, and I'll color this one in, why not? I'll shade it in quickly. Okay, so this is my conceptual diagram of a DNA molecule with sticky ends. So you can see these little overhanging regions. And these just aren't any old sticky ends. These are Hindi3 sticky ends. So Hindi3 sticky. Hindi3 sticky. And on each side, there's going to be a little sequence that we cut off that we're going to need to purify away from the longer fragment that we want to clone. And by clone, I mean put in the cloning vector and put the cloning vector into a bacterium. So these little pieces, we're going to get rid of those using another method that we can save for maybe biotech, BSc 353 or 354 if you take those classes. So, okay, so we have our digested DNA fragment, which we are going to call the insert, because we want to insert this fragment into a cloning vector. Now, we are also going to have to digest the cloning vector with the same restriction enzyme. And that's going to give us something that looks like this. Now let's see if I can conceptually diagram this in a way that's not too confusing. So the Hindi3 enzyme, if we choose the right cloning vector, we'll cut the cloning vector in the multiple cloning site only, and it will give us something like this. So, okay, I opened it up, and this is a double-stranded molecule, and I'm zooming in on the multiple cloning site here, and I'm showing you after digestion with the Hindi3 restriction endonuclease, we have Hindi3 sticky ends here. Okay, so once we have our digested cloning vector and our digested insert, both with Hindi3 sticky ends, we can mix these together in a test tube, and we can try to get this insert ligated into the multiple cloning site of the cloning vector. And for that, we're going to need to use DNA ligase. But let me diagram what that might look like. so we can all, I can make sure all of you understand how this process works. Okay, so one end of the vector I'm going to diagram over here. So here's one sticky end over here. Now, we covered what the sticky ends look like after Hindi 3 digested earlier in the video. And here I'm showing the base pairs getting smaller and smaller because I'm zooming in. I'm zooming into that sticky end of the cloning vector. So Hindi 3 sticky end over here. And then I'll put the other sticky end of the cloning vector over here. And I'll put the other bases over here, getting smaller. Okay, and then they're going off the page. So the cloning vector is about 3,000 base pairs long, and we don't need to diagram the whole circle for this particular part of the video. Now the insert, okay, we want to get that insert ligated into here. And remember, the insert also has Hindi 3 sticky ends. So let me put the insert like right here. I'll start with this end. And same thing, I'm zooming in on one of the sticky ends of the insert, and then I'll do the same thing over here. Put some ends in here because we don't care what those sequences are. Okay, so do you see what I'm doing here? I'm kind of just zooming in on the ends because what I want you to see is that these are complementary. So this A with that T, G with C, C with G, A with T right here. Let's see here. Do we have this right? What is going on here? Oh yeah, so that A goes with that T. This T goes with that A, CG, GC, AT. Okay. Now, on the 5' ends, there are going to be a phosphate. There are going to be phosphates. Let's see. And on the 3' ends, there are going to be hydroxyls, because that's the way Hindi 3 digest DNA. Okay, so now we're ready to add ligase. So if we add DNA ligase, what DNA ligase does is it takes hydroxyls and phosphates and makes links them covalently, forms covalent bonds between the oxygen and the phosphate. I should say, let me say that more precisely. Makes a covalent bond between the phosphate of the phosphate group and through oxygen to the adjacent nucleotide. Okay, so we're going to covalently link this insert into the vector with ligase. So we're going to get 4 covalent bonds, 1, 2, 3, 4. Okay, that's great. That's what we want to happen. The insert is now ligated to the vector. So that's one possibility, right? Because we're just mixing the inserts with the vector in a test tube with some water in there and some buffer for the ligase. But sometimes, and we hate when this happens, is sometimes this end of the vector can be ligated to this end of the vector with no insert. Okay, what does that look like? So no insert. And yeah, this is a pain when this happens. And there are ways we can prevent this from happening, but we're not going to cover those methods in this course because they're fairly technical. Okay, looks something like that. So essentially this, I can put the phosphates in here, phosphate, phosphate, hydroxyl, hydroxyl. Okay, so essentially this A is going to go with that T, G with C, C with G, T with A. Ligase is going to link those together. Ligase is going to link those together. Okay, so essentially the multiple cloning site was sealed back up. It's like we cut it with HindE3, opened up the multiple cloning site at the HindE3 site, added ligase, and then it just closed back in on itself without the insert. So there are two possibilities here. But one up here, the insert goes in, two, insert does not get in, and the multiple cloning site is reformed. It looks just like it looked before digestion with HindE3. So, okay, so what can we do? Now this is where that LAC-Z gene is going to come in. Now we talked about that earlier in the video. So we have two possibilities, vector with insert and vector alone. And I'm going to use this thing right here. This, you know, I figured I have such a hard time drawing circles. But hey, I have a genetics lab, and we've got these little petri dishes in there, these tiny ones. And this would be great for diagramming circles. This was an extra one that we couldn't use for media, so I'm not wasting anything. Okay, so vector plus insert, and what would that look like? Well, let's say this is where the insert is, and this is, you know, remnants of the multiple cloning site that we disrupted with the insert. Antibiotic resistance gene is over here. Same thing exists over here. And this is going to be the vector only without the insert. And here we have the multiple cloning site, okay, with no insert in there. We also need the origin of replication in both of these. And what is that for? So once we get these into a bacterium, the origin of replication will be used by the host replication machinery to replicate the plasmid. Now, there's something else in here, right? So this was, so let's say right here to right here, this is where that LAC-Z gene was in this particular cloning vector. And when the insert is in there, the LAC-Z gene is disrupted. So this plasmid no longer encodes a functional copy of the LAC-Z gene. This one over here, remember the multiple cloning site was integrated by, you know, scientists into the LAC-Z gene without disrupting or without mutating the gene. So here, the LAC-Z gene is still intact. So this plasmid right here without the insert, the vector only, the cloning vector alone, encodes a functional copy of the LAC-Z gene. And that functional copy of the LAC-Z gene here, but not here, is going to allow us to identify bacterial cells that possess the plasmid that contains the insert in the multiple cloning site relative to those that don't. It's going to help us distinguish between plasmids that have the insert, bacterial cells that have a plasmid that has the insert versus bacterial cells that just have the cloning vector alone. Sorry that I'm going back and forth between calling the cloning vector a plasmid and calling the cloning vector a vector, but it's the same thing to clarify. Okay, so let's see how we're going to take a look next at how this gene can allow us to select the bacterium containing the desired plasmid, the one with the insert.